Hello. I think I have never had such a wonderful introduction before. It's the coolest thing I've seen. So I'll try to live up to it. My name is Lise Arleth. As it was already told, I'm a professor in experimental biophysics in the X-ray and neutron science group at Niels Bohr Institute. What I'll talk about today is neutrons, X-rays, and shooting the unseeable biology. So, as it was already told in the introduction, we are building up this super duper microscope in Sweden right now. It's actually 2 microscopes, and here's some houses, and here's the city of Lund and Malmö, so they're quite big. It's a European Spallation Source, which is a big neutron scattering facility, and then right next to it we have a beautiful round building, which is a synchrotron, an X-ray synchrotron, so that's the Max 4 synchrotron that we are building up. And with these 2 facilities, we'll be able to look into many different tiny things. But the tiny things that I'm most interested in looking into are illustrated here. So this is a little, imagine that you could take a bit of your cell membrane and then cut it up in a little square like I've done here. Then you would see, they would not be red because they wouldn't have color on this length scale, but you would see some molecules that with this lipid bilayer here, they would have a little head group and some hydrocarbon chains, some oily chains, and that's actually what keeps your cell together. And then in this bilayer membrane here, you have all these proteins here, the green ones here. They are also not green in real life. They actually also don't have a color, but we have painted them green here. And these are the molecules that make sure that all the different molecules, that they can get in and out of the cell. And that's really important because all of us here, we are like many cell organisms, and we really appreciate that our cells are able to communicate with each other. And in order for the cells to be able to communicate with each other, then you need these membrane proteins. So they would be quite nice to understand. I'll actually start a totally different place because Niels Bohr Institute, you probably all know Niels Bohr Institute for theoretical physics. But we actually have a long and proud tradition for biophysics, and my personal hero here, this is this beautiful guy here, George de Hevesy. He was a Hungarian radiochemist, and a radiochemist, that's not a chemist that tried to make radios, but it's a chemist that investigates radioactivity, and there are not so many radiochemists any longer. But between 1900 and 1940, that was just the coolest thing to be, because that was when we had this radioactivity hype, and we tried, or the scientists then, they tried to investigate and develop radioactivity for all of those sorts of different purposes. This culminated with the development of the nuclear bomb at the Second World War, and that created some afterthoughts among many of the scientists, and so they tried to develop more peaceful applications of radioactivity. And one of them is nuclear power plants that some like, some don't like so much. But then also Hevesy, he was also one of the peaceful radiochemists, and he, first of all, he was the co-discoverer of element number 72, which he chose to call hafnium after Copenhagen. But what was really important of his work was actually that he decided that it could be interesting if he tried to water plants with radioactive substances, because I mean, who would think of that? But then the plants would suck up these substances, and then you could put a photographic film on top of the plants, and then you could see where different molecules, for example, phosphate or nitrate and so on, where do they go in plant, how fast, and what happens at all. And this actually gave him the Nobel Prize for his work on the use of isotopes, and radioactive isotopes mostly, as tracers in the study of chemical processes. So as such, he was actually the first biophysicist at Niels Bohr Institute. We that do biophysics at Niels Bohr Institute today, we don't do so much radiochemistry and radioactive tracers any longer, because that we did many years ago, and then we gave it on to the medical doctors and the biologists to apply, and now we want to do other things. And one of the big things that we are excited about these years, not only the biophysicists, but many of the physicists, this is this fantastic big equipment that we are building up in Lund, only 1 hour away from here. So there's actually 2 bits, as I said. It's the European Spallation Source, which is a neutron source, and I decided to put all the zeros on the price tag here today. It's expensive, but if you compare it to some of the airplanes that the military wants to buy now and so on, it's almost cheap. So, and I think we are many that will benefit maybe more positively from this facility than they'll benefit from the fighter planes. So I think it's fine to have this as well. Then right next to it, the Swedish government decided, that was slightly before the crisis had really set in, so they decided that they would top up and also build an X-ray synchrotron, so a really powerful X-ray source next to it. And with the combination of these two facilities, you can see that they're not really there yet. MAX 4 synchrotron will have its first light on midsummer in 2016, so that's actually quite soon. And the Swedes are very poetic, so of course it has to be on midsummer, then they turn on the light and so on. The European Spallation Source, I think they are happy if they have neutrons in plural in 2019. It's really a much harder job to produce neutrons than it is to produce X-rays. But we are working very, very hard on it. And when we get these facilities up and running in not so many years, then we'll have the world's best facility for neutron and X-ray-based structural investigations on sub-nanometer scale. That was a very long sentence, but it means that we can look into tiny, tiny, tiny details of all sorts of materials and substances with this super-duper microscope here, and that's really nice. So the questions that I will address today are the following. So first, as many of you probably have heard about X-ray investigations of all sorts of things. So you know that X-rays, they may be fine to look into materials, but why on earth do we also want to look into materials with neutrons? Why do we get such a crazy idea? In particular, if you know that neutrons, if you take neutrons, they live a nice and long life within the nuclei of the atoms. And they're very stable there, but if you pull them out of the nuclei, then they don't feel so well. Then they decay radioactively within 5 to 10 minutes, so they're really a bit troublesome to work with, and you also have to dig them out of the nuclear cores, and that's not so easy. And then also, if we want to look at something that is that small, then why do we need so big and also expensive equipment to see something that small? What is it really that we can see and how? And then why care about biological molecules, what they look like and how they work? Why care about that at all? I've actually decided to start with the end. I'll start with this part here, and then I will somehow work my way up here during the presentation here. Because I'll start with not really biological molecules, but with medicine molecules. And if you talk to some of the pharmacists and so on, then they basically talk about two different types of medical molecules. They have what they call small molecule-based drugs, and they are not very creative because a small molecule-based drug is basically a drug that is based on a small molecule. It's just not so many atoms that has been put together into a small molecule. There's a good example here of acetyl salicylic acid, which is the active substance in aspirin or idotyl, etc. And this is actually a molecule that has been known in popular medicine for hundreds of years. This is the extract of the bark from a willow tree, and it has been known for centuries that if you could extract this, then it would have like a nice sedating effect on you. By now, it's I think you can still extract it, of course, but it's much more economical, efficient to synthesize it chemically. So that's what big chemical companies like Bayer, for example, does. Then on the other hand, we have a more modern type of medical drug here. And this is the protein-based drugs. And again, a protein-based drug is a drug molecule that is based on a protein. Not very difficult. And the very good example of this, such a molecule, is insulin, which is crucial in the treatment of diabetes. And insulin is a much larger molecule than this one. I've not bothered to draw all the single atoms of it like I could over here. Instead, I have a whole chain of amino acids that are attached to each other here. And they fold up in a fantastic three-dimensional structure here. This here is actually not only one insulin molecule, but it's 6 insulin molecules. So it's a so-called insulin hexamer that is coordinated, that organize themselves around some central zinc molecules. When it was discovered that you could start to treat diabetic patients with insulin, that people that had diabetes actually lacked insulin and not just something else, then they decided, in the beginning they extracted this insulin from dogs or pigs, and that was of course feasible because we are more willing to sacrifice these animals than humans, of course, in order to extract insulin, but it took quite a lot of animals to save the poor diabetic patients. So around Second World War and after Second World War, it was found out how insulin could be produced from different other sources, and today it's produced by GMO-modified yeast, and that is really much nicer than killing a whole lot of dogs and pigs in order to produce this. So, but how do these molecules actually work, really? And this is what I'll try to explain here. So the small molecule drug that I started with, aspirin, it's a molecule that suppresses some enzymes, and the enzymes are really the molecular factories in our bodies. So enzymes are proteins that we have in our body, and they produce other little molecules. And if you take some aspirin, then this will suppress these COX enzymes. So they will stop producing so many molecules. And that will mean that they'll produce fewer of these signaling hormones that we need when we feel bad, but they will actually suppress signals about pain and a lot of other things. So this can actually be very nice. So if you feel a little bit of pain and so on, then or if you just want to have your signals suppressed in other ways, then it's actually nice to take an aspirin. then you'll have less information from your whole city, and or whole body, and that can sometimes be nice. With insulin, it's a signaling hormone. So this is actually one of those molecules up here that could be produced from the COX enzymes here, but it's a signaling hormone, and it actually starts a whole cascade. So it starts the glucose transport into our cells. I'll explain that more elaborately in a little while. And this makes sure that the cells, they receive the fuel that they need in order to convert these molecules, ADP to ATP. And the ATP is essential because that's the essential fuel for cells. So in order for cells to do all the jobs they have to do, they need some energy. And this is really this ATP that they rely on. And if there's no insulin, then this cascade doesn't work. So this is 100% essential for life, that you get this cascade going. So this is a very specific, in contrast to aspirin, it's a very specific interaction that we have here. Then I have a third type here, paclitaxel, which is a cytotoxin, in Danish, a cellegift. And also the chemist or the pharmacist, they're actually not so, I mean, you think that they would be geniuses, but they go and take off bark from different trees, and they do extracts and plants and so on. And then they see whether it works and how it works and so on. And paclitaxel, which is a cytotoxin that is used extensively in cancer treatment, it's one of those such molecules here. So it's extract from bark of yew, which is called tax in Danish, and it kills cancer cells, and unfortunately it also kills all other quickly growing cells. So that means that the molecule is very, very unspecific when it works. So on one hand we have the protein-based drugs, which I think will be the future of the drugs because they are really specific. They can go in and resolve a specific function. And then we have these small molecule drugs like aspirin and paclitaxel and so on, which are very unspecific but very cheap to produce drugs. But how do the medical molecules actually work? So now I told you how they feel and a little bit about the enzymes and how they dampen and all that. How do they really work? And in order to understand that, we have to first zoom in. And here's a picture of a typical mammal cell. So a cell that could be from one of you, maybe, probably not, but a typical cell here with a cell core in here. The insulin receptor, it sits out on the outer plasma membrane up here. So such a cell here, it's about 5 micrometers. So it's something that you can actually see in a microscope. Then these insulin molecules here, they sit out in the outer cell membrane here, and when we look at how insulin works, then first the insulin molecule comes and binds to this receptor. This receptor is a membrane protein. So one of the guys that I told you in the beginning that I really like because they are so important, insulin goes down and binds here. then this one somehow changes conformation down here, such that a whole signal cascade is started, such that some little depots of glucose transporters that are near the surface here, they somehow get the information that they should go up to the surface, merge with the surface, unfold up here, and then open up for the glucose transport into the cell. This is actually quite fantastic that little insulin can take care of all this. But if we look at the length scales that are involved with this, then the thickness of this cell membrane here, it's only about 5 nanometers. So that is 5 times 10 to the minus 9th meters, or 1 meter divided by 1 billion. It's not a lot. It's a very, very short distance here. So the good question to ask is, how do we know all this? When I make such a drawing here, then how do I know? Because it's clearly not something that we can see. And how do we know what the molecules look like at all? And this is actually... So now we are at Christiania, and I think it's suitable now that I take you to a very special place. And this place is called Reciprocal space. And it's it's not like any other spaces that you have been to, I hope. So Reciprocal space is a place where long distances become converted into something very short, and short distances become converted into something very long. And then try to imagine what happens in there. I think if you're a physicist doing X-ray and neutron scattering, then you think reciprocal space is a quite cool place to be. And as one of my former PhD students once said that, reciprocal space, that's actually the place where all physicists would like to go when they die. That could be really nice. So, and then I hired him straight after because I think that was so cool. But reciprocal space, that's actually something we need in order to find out how these very, very tiny things look like. And we can get to reciprocal space by shining either X-rays or neutrons into very small molecules. Here there's a very nice picture, and very famous picture also. This is the first X-ray diffraction pattern that was really used for obtaining big insight into science. This is the X-ray diffraction pattern that was obtained by Rosalind Franklin in the 50s of the DNA structure. This picture was then used to calculate backwards from these beautiful pictures here, where you have all the large distances about the DNA. They will be placed close to the middle here, while all the short information, they will go out here towards the sides here. Watson and Crick, from this picture, they could actually find out what the DNA structure looked like, and gave us this superhelix model here that we now is now quite important. At least it's quite important that we know how it looks like, because then we can much better understand how it works. So Watson and Crick received the Nobel Prize for this, and Rosalind Franklin, which was a woman and in the 50s and so on, I think, no, she'd say, I'm just taking this picture, this was nothing. So she didn't receive any prizes for that. But I think she would have received that today, and she has received lots of the honor afterwards. But in order to obtain such a picture here, you have to have a crystal of the DNA. So this superhelix structure here, you have to crystallize it somehow. And this is, of course, a tremendous task. You can crystallize salt or sugar and so on, but imagine that you had to crystallize a big floppy molecule. That is somewhat harder to imagine, but this can actually be done. And if you obtain such a crystal of, for example, DNA or of a protein, then you can take this crystalline solid, then you can take an X-ray tube, and then you can turn it on. And then if you place, here's a LED screen that is placed after the X-ray tube, where you drill a tiny hole in it so that you get a very well-focused X-ray beam out. Then the X-ray beam comes out, it interacts with the crystal structure here, and then you get this nice diffraction pattern out. And here we are in reciprocal space, so you can see this is really, really beautiful. Nice spots, and they sit in a symmetric pattern. And from this beautiful symmetric pattern here, you can calculate backwards and find out what the crystal looks like. And this is actually how we obtain this insight, and there are no other ways of looking into so small structures than by sending X-rays into it and then going a tour in reciprocal space and then hopefully coming back to real space and find out what the molecules look like. So the big advantage of X-ray crystallography, which is really the main technique in the field, is that it has a structural resolution down to 1 angstrom. So this means that we can get atomic resolution. We can see where the single atoms sit in the molecules, and that's quite something. The big disadvantage is that it requires that you can crystallize your molecule, and most of the really interesting biological molecules, they are sort of not very quiet, and they don't want to lie down in a big crystal with 10 to the 15 other molecules and stack up with them in a totally coherent way. They actually like to move much more, and you need some other techniques to investigate those molecules. And alternative techniques, there are several alternative techniques. My technique here, which I of course favor because I think it's the coolest technique, but there are many techniques, and this is called Smallangle scattering. And like in crystallography, you take a beam, this can be a neutron beam, and then the technique is called Smallangle neutron scattering, which is abbreviated SANS, or it can be an X-ray beam, and then it's called Smallangle X-ray scattering, SAX. But then you take a vial of your sample, and here you can see that I have taken some worm-like particles that just swim around and so on, and I just put them into the beam, and then I obtain a beautiful scattering pattern in reciprocal space over here. So this is quite nice, I think. As you maybe remember from before, if you had a crystal that you put into here, you would obtain some nice dots. I don't obtain those nice dots in my pictures, unfortunately. I obtain some rings, which can also be nice, but they don't contain as much information. But the good thing is that then I don't have to crystallize my sample, so I can just put anything into my sample. So some years ago, I took a piece of a car tire that I just put in there, obtained beautiful scattering patterns, and you could actually see what a car tire material looks like down to a nanometer scale. If you're a car tire producer, then that's interesting. So the big advantage of Smallangle scattering is that you can measure all samples. You can just put anything into the beam. The disadvantage is then, because you have all this flexibility in the sample and they don't do the same movements at the same time, then you can only go down to about 1 nanometer or 10 angstroms. This is still quite small, but it's not atomic structure, so we can see tiny things, but not tiny, tiny things with this technique here. Then I already told you, but I'll explain it again here, this travel to reciprocal space. Because if I have an insulin molecule, and then I Fourier transform it, or I do a scattering experiment on it, then I'll obtain this scattering pattern where all the information in this picture over here, so all the information about the large distances on the insulin, so for example, what the diameter is, that I will find in the inner part of the curve here, because large things become small. But then all the tiny details about the distance from the coils here and so on, that'll be available out in the outer rims of this picture. So I have to get my head tuned into thinking everything a little bit upside down. But when I do that, then I can actually find out what this molecule looks like from this picture over here. And here I have actually drawn a molecule that I know what looks like, so that's not so interesting, because why on earth all the trouble? But in many cases we don't know what the molecules look like, and then it's really important that we have a technique to looking into it. Then I already mentioned the word Fourier transform, and this is also something that we as physicists are quite happy with. I mean, not that it's called a Fourier transform, but we are happy that we know how the two are related to each other. So we can actually calculate, if we know what this one looks like, then we can also calculate what this one should look like more or less, and sometimes we can even go the other way back, and that's really good. So many of the old crystallography pictures, they have been taken by X-rays, but I also mentioned that we are building up European Spallation Source, which is a neutron source, and I told you that, and you already know that, I guess also, that neutrons, they are particles that sit in the nuclei of the atoms. And how on earth can we take some particles and then obtain diffraction patterns? Because diffraction patterns, you're probably used to thinking about that has something to do with light and light waves, something more continuous and so on. So, and here there's actually another question that they discussed a lot at the Niels Bohr Institute and in the rest of the world in the first half of the last century, and that is the particle-wave duality, that particles, they can both be particle-like and they can be wave-like, so they can have both types of behavior. So neutrons, they are particles, but if I take a whole neutron beam and send it off with a certain velocity, then it'll actually behave like a wave. And it turns out that we can even relate the two. We can relate the velocity of the neutrons to the wavelength of this wave, which I have called a lambda here. So if I have a neutron of a certain velocity, v, then I can calculate it simply from this equation here, where h is Planck's constant, which has it always turns up in all physics equations. We almost always have something with Planck's constant. So this is also present here. And then m here, this is the mass of the neutron, just the rest mass. And then you can relate the two. So you can easily relate these two to each other. So the neutrons, they are a little bit, like those sensitive men that were modern a few years ago, which, didn't really know whether they want to be macho or didn't really know whether they want to be, more feminine and in touch with their emotions and so on. And with the neutrons, it's a little bit the same. They actually don't really know whether they want to be a particle or a wave. And if we have to, if we as physicists have to manipulate these neutrons, then we have to treat them with care. And when they want to be treated like waves, then we have to do that. If they want to be treated as particles, then we have to do that. And they don't really tell, and we just have to. But so it's a little bit troublesome, But we like them, and we like them just the way they are. They also help us in many ways. So it's good to have them. But unfortunately, they're very difficult to produce. So how do we generate a whole beam of neutrons? Well, the normal way is that you build a nuclear reactor. And that is like a big equipment. We have a few nuclear power plants in Sweden. One we can see from Denmark, and we don't have any longer in Denmark. So it's big equipment because you need to run a controlled fission process in order to produce neutrons. You have to drill them out of the cores. So you have these uranium-235. And you can excite it. If you already have one neutron, you can excite it to uranium-236, which is unstable. And then it falls apart, generates 3 new neutrons, which can then enter again. And then you can produce more and more. If you don't control this, then you have a nuclear bomb. If you control it, then you have a nice energy-producing nuclear power plant. And of course, there's a risk associated because it's so close to the bomb as well. So in Sweden, and when we started to work for European Spallation Source, it was already, so the politicians and the society was already not so happy with the nuclear power plants and nuclear energy and so on. So we decided that we had to produce these neutrons at the European Spallation Source in a more, I will not call it an environmentally friendly way, but in a less dangerous way. And this is why we decided to aim for the spallation process. And in the spallation process, you take a heavy metal target, and then you bombard it with protons, and then you excite these target molecules here, and then they basically start to sweat off the neutrons. Then the neutrons will fall off, and then you can harvest the neutrons from this target here. ESS is based on this spallation process, and at ESS that we're building up in Sweden right now, it's the protons from a 300 meter long, 5 megawatt linear accelerator, and those protons, we bombard them into a rotating tungsten target that is a couple of meters in diameter. So we have this big disk of tungsten, which then is rotated, and then we shoot these protons into it, and then there's a lot of neutrons generated that we harvest. And spallation, that's actually not really a good Danish word for it. I think the closest thing I could come to would be in Danish when you talk about something where you say at man slår flint af noget. And this is actually also what we in a way do here, that we slår flint, neutronflinter of these cores here. So I think if we should find a good Danish name for European Spallation Source, it should be the European flintekilde, eller den europæiske flintekilde. Jeg ved ikke, om det lyder ikke særlig high-tech, så måske passer det meget godt med tiden og sådan noget. Og så kan vi jo altid gemme alt det her fantastiske oppe bag navnet her. Now I change into Danish, and I'll swap back to English. So I told you that the neutrons, they are hard to manipulate, and we have to, treat them with care and so on. They're also not so easy to produce. Either a fission reactor or a spallation source, which is both quite expensive and big. So why on Earth, when we can do all these wonderful things with X-rays, why on Earth going through all the trouble of producing the neutrons? And to illustrate this, I've taken this drawing here, so we don't actually know whether it's so big and whether it really looks like this, but this is what I imagine it looks like. And I have a membrane protein that sits in a cell membrane. If I look at that with X-rays, then I would get a nice structure out where I would see both the membrane protein and the head groups of this lipid bilayer here and the tail groups, and they would have different contrasts. That looks nice when I look at it in direct space, but when I look at it in reciprocal space, it unfortunately looks less nice. It's actually difficult to interpret that picture. The huge advantage of neutrons is that we can play with the contrast and we can change the contrast as we go along. So if I just take the same system and instead of normally H2O-based solvent, then I use D2O-based solvent, then I will have this membrane protein here, and then I will have only a two-color system because I'll not see the difference between the heads and the tails here. This sounds like I'm getting less information now, but maybe I'm zooming in on some of the exciting information, and that can be really helpful. Then I can play another trick, because I can actually go ahead and deuterate the membrane here. I've still drawn it here, but you can actually turn down the signal from this membrane so much that you only see the membrane protein. So then you have a really nice picture of, if you want to investigate the membrane protein, then it's nice to see this and not all the blur and membrane around it. Then there are a lot of people that actually think that much of the secret of these membrane protein that actually also lies in the surroundings, in the membranes that surround them. So here with the neutrons, you can go to a third contrast situation where if you mix H2O and D2O, then you can actually mix it in such a way that the protein here becomes invisible. And then suddenly you can only see the membrane here. And this is really the cool thing about neutrons, that you can change contrast, play with the contrast systematically. And with X-rays, we have lots of X-rays, so that's really nice, but we cannot control their contrast in any way. So that's why we bother to work with those complicated neutrons anyway. So if you do neutron and X-ray scattering, you rely on these big facilities. We don't have a nuclear power plant at the University of Copenhagen. I'm almost laughing because it's totally ridiculous to think about that we would have a nuclear power plant in Denmark by now. We also don't. We have a small synchrotron in Aarhus, but that doesn't produce X-rays with the right energies. So actually the place that my group likes to go to is Grenoble in France, which lies in a nice valley, and in this valley you have this Institut Langevin, which is in this container here. There's a nuclear reactor, and so here there are a lot of neutrons are produced there in the core of this reactor, and then they have holes and drill holes into the reactor, and then they can harvest neutrons from all sides and use them for experiments. Right next to it we have the European Synchrotron radiation facility, which is this big ring here where we can use to generate X-rays, and then we also harvest these X-rays and use them for a lot of different types of experiments. There's several excellent facilities, different places in Europe. There are not so many neutron facilities, but there are actually several really good X-ray facilities, different places. So we also use the other ones, but Grenoble, I think, is the favorite spot right now. So these are large-scale facilities. They're, of course, really expensive. So if you want to do experiments there, then you really have to use the time with care because it's precious. So as scientists, we compete for beam time, as we call it, at these facilities. So we submit an application, explain what we would like to do, and then if we are lucky, we get 24 hours of beam time. And then, there's this photo here where we actually went a large team there. We had 24 hours of neutrons first, and then 24 hours of X-rays afterwards. So 48 hours in a row. And then we had to be a quite large team in order to exploit that optimally, but it normally goes quite well. This photo is taken after the session. People are still sort of smiling and eating, and I think most of them went to bed right after. It was quite tough, but it's a fun way of working, I think, and it's really almost like going on an expedition, except that we have all the samples from home, and then we go and harvest all data, so it's slightly different, but I think it's fun. So now I'll be 4 slides of science, and it'll real science. So the big advantage of small-angled scattering is that we can actually predict what our data look like. So if I had a test tube of little spherical particles of 60 angstroms, then I can calculate that my small-angle scattering data would look exactly like this. That's really nice. If I had rod-like particles, the scattering pattern would look like this. If I had worm-like particles, as over here, it would look somewhat like this. If you're trained in looking at reciprocal space pictures, then you think that these pictures look dramatically different from each other. It's very easy to see the difference. If you're not so trained in looking at those pictures, it just looks like, decaying curve with oscillations and so on. But this is actually quite nice. The big problem with small-angle scattering is then that, it's also illustrated here. What we go and harvest when we do an experiment is that we get a scattering pattern that in 2 dimensions looks like something like this. Beautiful. We would love to be able to press a button and then have a computer doing some calculations for us and then hopefully find out that our molecules look like this. Unfortunately, we cannot do that with small-angle scattering or with any of the other scattering techniques. We have to go through some math, and we can only go this way here. So we can only go the other way where we say, okay, I now assume that my molecule looks like, for example, this here. Then I have to develop a mathematical model for this. This is what I've illustrated down here. Then I have to take into account that I cannot measure an amplitude, I can only measure intensity, so I have to take the square of the whole thing. This is what is illustrated here. Then I also have to take into account that if I have 10 to the 16th of these molecules in my test tube, then they'll have all sorts of different orientations. So I also have to build that into my model. And this is what I've illustrated with these brackets here. And then I can calculate what my scattering pattern would look like here. But this is unfortunately both a so-called inverse and a non-linear, and therefore a quite complex problem. So if I have to be honest and tell you what I really do most of the time when I'm at work, then it's actually something like this here. So the secret weapon that we really have as physicists, and I hope that you cannot read the equations here, so at least I'll not go through them. The secret weapon that we have as physicists that we really rely heavily on through all our science is that we are really, really good at setting up these equations and programming it and have the computers work hard to crack our structures for us. This slide here is from one of my former PhD students, Nicholas, and he actually tried to make a really communication-friendly slide to illustrate what it really is that we are doing. And I know he's very nice and a slightly nerdy physicist, so this is what communication looks like when you're a first-year PhD student. He graduated and he has had a nice career afterwards. So what can we use this technique for? Now I'm back to one of the applications here, and here I'll also reveal one of the big secrets, not a secret, but one of the big things, the core businesses of one of the large companies that we have here in Denmark. So if you have a protein-based drug that I talked about earlier, and that could, for example, be an insulin hexamer, Then when you have to inject it, so normally with proteins you're used to looking at these declarations of food, and you can see that the food maybe contains, 5% proteins and so on. But those proteins you just eat, and then they are digested in your stomach, cut up into amino acids, and then those amino acids go out in your body and you use them to build new things from. So that's just building blocks for new things. But if you really want insulin and you eat it, then you'll not get much out of it. So in order to get any effect out of insulin, you have to inject it subcutaneously, so right under the skin, from a water-based solution. If you're a drug-producing company that wants to sell insulin, you normally have to be able to prove that you can put these molecules, a lot of these molecules, into a vial, and then the customer can place them, or the pharmacy can place them on a shelf for up to 2 years, and maybe in a third world country where the fridge breaks down now and then, and so on. So you have to be safe after 2 years also. That's a challenge. And then the hard bit comes, because then when you inject the insulin, you would also like to control how fast it works. Because if you're a diabetic patient, it's not very nice if you have to go and inject all the time. So it would be nice if you only had to inject maybe once per day or once per week or something like this. So that's why the big drug-producing companies, they put a lot of research into controlling what is called the release profile. So when you inject the drug, then how fast does it distribute itself? And the original types of insulin, they are sort of medium-releasing drugs, and that's basically the hexamers that I also showed you up here. And in order for this to work, you inject it as a hexamer, and then the hexamer wanders out in the body, and then it falls apart into dimers, which are these green ovals here, and then these have to break up into monomers, and then the insulin cascade can start. So this slightly released, or slightly decayed release here. Delayed release, sorry. If you want a fast release, not surprising, you surface modify the molecule here slightly so that it doesn't like to produce these hexamers any longer, but instead it enters your body as dimers, and then these dimers only have to fall apart into monomers, then they work. And then if you want to have a slow releasing molecule, then you can put basically some glue on the outside, some molecular glue probably, that sounds more cool. But anyway, something that makes the insulin stick to each other. And then they form these large complexes of hexameric insulin, which is basically some nano-sized depots that forms right under your skin when you inject it. Then these hexamers have to fall off first, and then the hexamers again have to fall apart into dimers, which then have to break up into monomers, and then you have the effect of the insulin. So that's a slow release, and if you can inject a large depot of this under your skin, then you don't have to inject insulin so often. So if you're a company that produces these kind of things, then it's quite essential to understand and control how such a protein can both be stabilized for storage, but also how it behaves when injected. And if you can do this, then you are in for really, really big business here. I think it's $1 billion industry to be able to control this. So if you can design some molecular glue that can control this and secure that it's released smoothly, then you don't have to do much more in your life, I think. We tried in our own humble approach to investigate some of to find out what the secret of some of the molecules from Novo Nordisk was, and we took some, and here you have some real small angle scattering data that I have measured myself, actually. So it's insulin. There was varying amounts of glue in the molecules. There was also varying amounts of salt in the molecules. So there's two series of data. There's one where we had 30 millimolar salt, and this is basically the amount of salt that we have in the vials before injection. And then one where we have 150 millimolar salt, and this is the salinity, the salt concentration that we have in the blood. And you can see when we look at the molecules in the vials, then they are actually hexameric or dihexameric, so they're quite small, which is nice if you want to keep them stable. Then it's good that it's always dangerous if you have something that's too big, then it also wants to form something that is even bigger and so on. But this was actually quite small. You could see that they changed from being hexameric to dihexameric as we put more glue on them. But then when we changed the salt concentration to the levels in blood, then we could see that still if we have no glue on, they would still be hexamers, but then they would form these long rods, which actually is the same principle that also is used in the most modern types of long-lasting insulin. That is they form these long rods, and then these hexameres probably falls off from the ends and then off to these hexameres, and then the release starts. So that is actually, we could directly see that from the small angle scattering. Of course, we could not tell Novo Nordisk much new here. They kind of already knew this or something like this. So we have to dig a little bit deeper into that. But what would we like to, when we get ESS and Max 4, because this we could already do with existing facilities, what would we like to be able to do much better in the future? There are many, many questions that we would like to investigate with ESS and Max 4. But one thing is that, general thing is that we would like to obtain a better understanding of nanoscale structures in biology. I told you and explained this insulin cascade, which is crucial for life, to you before. We want to find out what it really, I mean, this is just a drawing, and many of the things we know, many of the things we don't know, we would actually like to see whether it really looks like this. Another great example is this so-called G-protein coupled receptor, which is a really important drug target, where the structure of this was determined a few years ago. This also gave a Nobel Prize to the scientists behind it. It's quite important to know what these molecules look like, because membrane proteins, they are the molecular target for about half of all medicine. And the big problem is, of course, that the membrane proteins, they're really small. They're only about 50 angstroms or 5 nanometers. And then they sit in this membrane, and they like to change conformation and so on. And it's actually extremely difficult to study while they sit in the membrane and while they do their job. So that means that we know extremely little about membrane proteins. And it also means that all the modern methods we have for developing new drugs, they're basically based on trial and error rather than intelligence, I would say. When the big pharmaceutical companies, they don't like the word trial and error because it sounds like they don't really know what they're doing. So they say that they use high throughput methods and so on. But high throughput methods is just another way of doing a lot of high trial and error in parallel. So this is actually the way that they go about now. It could be nice if we could actually understand some of those molecules better, then we would probably also be able to do more intelligent development of new drugs. And then my personal grand goal is, because now, I mean, it's nice to know what they look like on a picture of this. Actually, I would really like to find out what they look like when they function. So when the molecule comes and binds, they change conformation and so on. How can we see that? So I would really like to be able to obtain a molecular movie of biological molecules in action. I think that's one thing that will probably keep me occupied the rest of my career. It's not particularly easy, unfortunately. So all this, we hope, all these answers, we hope that we'll be able to probably not close an answer, but start to address with ESS and Max 4 and some of the other nice facilities that are coming up. But in the beginning and in the next many years, at least when they come up, these will be the big facilities and will really be the best in the world. So I think in 5 to 10 years, I will not go so much to Grenoble any longer, but I'll go a whole lot to Sweden and hopefully get a lot of beam time there. So. And then I'll end by asking the question, now I have told you a lot about medical molecules and biological molecules, and I come from a physics institute, so it's all this physicist, and what do I tell the people that employ me and so on. But I say that, yes, I think it's physics, because we heavily rely on methods from physics to investigate the molecules. And also, I think, just like Hevesy, I think we are much more interested in finding out how to solve a problem than actually in using it for a whole range of biological problems. So as physicists, we like to think of ourselves a little bit as pioneers. We try to move out in a new area, find out, now we can do this and that and so on. And then when we have done it 2 or 3 times, then we don't need to do more experiments there. Then we like to go somewhere else. But then hopefully some of the most important results, they will be picked up by biologists and engineers and so on, and be used to investigate a broad range of different molecules. Hevesy did very much the same. He developed a technique, used it a few times, then went on to something else. I think I will do exactly the same as physicists. That's kind of encoded in our heads. But as physicists, we depend very strongly on collaborations with biologists and medical doctors in order to address these questions, and in particular to secure that it's the relevant questions that we ask, and not just a nice question that we can solve with some big toys in Sweden, but that we can actually help contributing to society and give back from what we have received. So concluding remarks, I hope that I have convinced you that many problems of biological or medical character, they actually need methods from physics and also physicists to be solved. Then also, for all future science, it's essential with interdisciplinary collaborations. There are few, I think if you're really a hardcore theoretical physicist, then you'll probably mostly talk to other physicists. But many of us that investigate real samples from the real world and so on, we are really highly dependent on being able to collaborate with, for example, with biologists in order to produce our samples and in order to ask the right questions. And then I also hope that I have convinced you that there are some really, really interesting perspectives with ESS and Max 4 coming up in the neighborhood. I think a large fraction of the physicists that we now train at the Niels Bohr Institute, they will probably end up in some relation with those two facilities. And I hope that that'll also count for many others. So with this, I would like to thank you all for the attention here. It's been nice to have you all sitting here. So thank you.